Hello, everyone, and welcome to our webinar today. Let me I'm going to start by introducing all of the panelists. Okay, so my name is Sergio Bacallado, and I'm a mathematician at Cambridge, and I'm also an admissions tutor at Magdalene College. So Magdalene, spelled Magdalene, we pronounce it in this weird way. We have a very special panel today. So we have Professor Franklin Aigbirhio. Maybe Franklin, you can say hi. And
hello, everyone. Good to meet you.
Alexander Olive from Target Oxbridge.
Hi, everyone.
And we have two very special panelists who are undergraduate students in Cambridge. So Ethan.
Hello.
And Ella.
Hi, everybody.
Okay, so you'll get a chance to hear from all of them a bit more. But in the beginning of our session today, Professor Aigbirhio is going to give a talk. And so I just want to give a very brief introduction so you can know a bit more about who Franklin Aigbirhio is. So Franklin he started his education at the University of East Anglia in Norwich, where he obtained a bachelor's degree in chemistry in 1984. And from there, he moved on to the University of Sussex, where he did a doctorate in chemistry. And after that, in 1988, he joined the MRC, the Medical Research Council, where he started a very distinguished career in chemistry. And he currently holds the position of Professor of Molecular Imaging Chemistry at Cambridge. So this is the highest academic rank in Cambridge, that of professor. He's also the director of PET radiochemistry at the Wolfson Brain Imaging Center, which is an international reference center for medical imaging. And he has earned many accolades. He's a fellow of the Royal Society of Chemistry. He's a senior research fellow in Magdalene College, which is why he's giving a talk in this event. I've had the pleasure of working with Franklin as a collaborator, and he's a global leader in the field of PET radiochemistry. So here, PET, I should say, does not refer to cats and dogs. It is an acronym that stands for positron emission tomography, and hopefully you'll learn much more about it from Franklin. But he has, just to give you some statistics, Franklin has authored over 300 research papers, according to Google Scholar. And he's responsible for bringing, just in the last decade or so, more than 12 million pounds in research funding to Cambridge. So he's an extremely eminent chemist, and we are very honored to have him give us a presentation about chemistry and medicine. Okay, so thank you very much, Franklin, and you're welcome to start your presentation.
Well, thank you very much, Sergio. I recognize myself there. And thank you, Target Oxbridge. It's a pleasure to give this talk. And something kind of really kind of passionate about in terms of increasing BME entry in the university. One of the things that which probably maybe didn't mention, I've recently become the co-chair of the university's BME staff network, and really to try and address in terms of various things, in terms of course, in terms of attainment of BME applicants, but of course clearly in terms of admissions. So I think I'll share my talk, and I think hopefully it'll come out okay. Share, and I'll go into mode, and hopefully slide mode. I don't know which one you see. Are you seeing just the slide as properly? Okay, because he always has two versions, which I'm not quite sure. That's the kind of the presenter version and the viewer's version. So as I say, I'm molecular imaging chemistry here at the university, and a fellow of Magdalene College, and also a co-chair of the BME staff work. So what I'll try and give you is a kind of flavor of, as I'm originally, I'm a chemist, and I'll kind of take you through a bit of how I got here. And here being, in essence, where I am, and it's as I'm based really here on the Cambridge Biomedical campus, which is on the southern side of Cambridge. And I've really been in Cambridge for now gone to 25 years. And when I kind of came here, it was just Admissions Hospital, and it is now this huge campus. That's its largest biomedical cluster in Europe, with the obviously university, like many kind of combination of medical school and university kind of intertwined with the university medical school there, which I'm part of, and of course Addenbrooke Hospital itself. And then if I can use a pointer, can you see a pointer on this? And can you see a pointer?
I don't think that we can see your cursor.
Okay. Oh no. Okay. All right. Well, okay. Never mind. So I said campus with kind of Addenbrooke Hospital, the Papworth Hospital, AstraZeneca, and of course the MRC laboratory from Microbiology, which you maybe have heard of. And where I'm actually based myself is in the Wolfson Brain Imaging Centre, which I'll come to very soon in what it is. But of course, it's clearly a really great place to work in terms of which I suppose my underlying theme of being a chemist, but really important part of directly involved in biomedical research. And this is a real good place to really interface with that, and really interface with many of the different groups, from clinicians, biologists, physicists, to really in terms of understanding disease, and of course translating it and actually making an impact in terms of patients. So really how I got here, and in a way, as kind of Sergio touched on, I got my original degree in chemical science not far from Cambridge at Norwich, East Anglia, and then did a physical organic chemistry PhD, and then organometallic chemistry, and of course, and then from then, in essence, I won't go through the different hoops. I ended up here at the Wolfson Brain Imaging Centre, which is actually the Department of Clinical Neurosciences, and really from on the basis, I can see of doing bench-side type chemistry, the usual kind of test tubes, clinical flask and such. I really find myself in this kind of situation of really being very much in terms of really in a clinical setting, of really in terms of and this underlying theme of my talk, of really applying chemistry for biomedical research. And the unique thing about the Wolfson Brain Centre, it really is directly applying. And in this case, we're doing in the very early phases when I first arrived here, and the focus was to studies on acute brain injury patients, and using techniques which I'll come to. So it really is kind of I suppose in terms of what I've moved from really, but was bench-side chemistry applications to really in terms of directly into working with patients, and that's really something I truly enjoy. And what I probably try and give you is kind of flavor of the kind of techniques which and the sciences I bring into, but of course the core of it is still very much of being a chemist and applying chemistry to what I do. So what I'm involved in is molecular imaging, which is a very new term, I suppose, a most better term for what we do. And it's really, I suppose, to kind of capture what's happened over the last, I suppose, 30 years of imaging, which is applied for, in terms of, patients, applications, or even kind of more, I suppose, biomedical research. And of course, once you're probably more used to, and of course, is computer tomography, which is very much against identifying structural, and you probably see more what I keep on showing in terms of images with the brain. My focus tends to be on brain imaging, but I might apply imaging to other parts of the body. But by definition, I've been based at the Wolfson Brain Imaging Centre. Brain imaging is part of focus. And here's a snapshot of the brain by computer tomography, kind of sliced across the brain, shows all the. It'd be good I can get some the marker to work, but never mind. But that shows very much the structure, and that's where you could say it's been the primary strength of imaging for over the years. Of course, a more powerful method for actually structural characterization of the human body in vivo is MRI. And of course, that's more, again, widely known as, many hospitals have this MRI, so a truck might come in or based. And it's a technique which is clearly powerful, and as we say here, also in Wilson, we have used that intensively for, obviously, for clinical diagnosis, but also for research diagnosis, and we are using far more powerful techniques to really to get even more higher resolution down into millimeters, sub-millimeter aspect. Well, I suppose is my speciality has really been what it calls positron emission tomography, and I'll put that into detail. And here you can see it's a bit more of a, less resolution you'll see here, but what we can give, which is unique in that, is giving you the, I suppose, for a better word, the biochemistry of the brain. Rather than structural, is the chemical aspects, the enzymes and all the other things present in the brain or the other parts in the human body, which you can't really determine from the more structural focused techniques of MRI and computer tomography. So and of course, with those techniques being around, the real strength more recently, of course, is now starting to merge those techniques, because they have strengths and weaknesses. As you probably saw in the PET, is the resolution it's not as great. It's probably down to 2 or 3 millimeters, while it got a very high resolution of MRI. And so what we've come more recently, only in the last, I suppose, 8 or 10 years, is combining the techniques, bringing in the MRI with a high resolution of brain imaging structure, and this very much nuclear medicine technique of PET to then kind of merge two kind of things. Let's see if I should almost find a way, laser pointer. Right, and I think that's it, found it to work. So here's basically the MRI, the structure. Here's the PET, given the biochemical information. And what we can do now is we can merge it, do what we call co-register, where we actually put the structural information to map onto the biochemical information and really get a more rounded information of the body and characterizing the body, not just in structure, but in biochemistry. And that's a real powerful technique in terms of really applying for biomedical research, for clinical diagnosis, and of course, in terms of potentially developing therapies. And this is where the techniques which we are very much using and where we are going to in terms of the where we are in the Wilson. So really, I've heard it mentioned several times, positron emission tomography. I'll kind of break that down exactly what that means. So basically, in essence, what will consist of is, in essence, injecting into human a solution of a chemical form which can distribute in the body and such. But of course, the unique thing about this chemical form, it has a kind of, it decays in a way that gives a positron, hence the positron emission, and in a way, but that doesn't really travel very far. At most, in the human body, kind of 1 or 2 millimeters, and that really collides with the electron, and that gives 2 gamma rays in coincidence. And that's the real power of it. Those 2 gamma rays can be picked up by high resolution, high sensitive scanners, and then we can use that to actually determine, in a way, indirectly, where that distribution of that molecular probe is in the body or in the brain. And then we can then use that to not only just obtain the distribution, but obtain the quantity. And from that, actually know from this part of the brain, in essence, what it represents in different amounts of the chemicals. So here's and of course here's I suppose a kind of a visual representation of what we will get from that scanner. As I mentioned, the resolution is not the highest in terms of the different types of techniques. 1 or 2 millimeters can be obtained and also 3 to 8 millimeters in clinical. But the unique thing, of course, is real high sensitivity. So we can go down to kind of picomolar levels and actually quantify very small amounts of enzymes, receptors, and biochemical processes in the brain. And that's a real power for sensitive determination of variation of a disease pathology. And of course, underpinning this is, of course, is chemistry. So, but the unique thing and the challenge, and of course the difficulty that makes it really almost like high-end type of science is that we are dealing with what we call kind of short-lived radioisotopes, so unlike you're probably used to the more longer-lived of carbon-14, which of hundreds of years or whatever. These forms, which are less commonly encountered, are very short-lived. So carbon-12, which we know, stable carbon, which is kind of permanent stable, the variation of that is a carbon-11, and that has a half-life of 20 minutes. So therefore, every 20 minutes, half in terms of the activity. So really, you can usually say you generate it in the morning, by the afternoon, completely gone. Other much shorter form, a form of nitrogen, 13, 10 minutes half-life, a variation of oxygen, 2-minute half-life, and of course a bit much longer, which you quite like, a 2-hour half-life. So they are very short half-life radioisotopes, which have to be used on the day of creation. And so to do that, and really to able to use these things, we can't import it from anywhere. So at the Wilson, underneath our kind of our main building, we have what is called a kind of cyclotron. And in terms of size, just to put you in context, if someone was standing, they'll probably be standing there. So it's quite a relatively compact system. But this is actually open. You can see the inside. Normally it'd be closed. So what this cyclotron can do, and it gives its ability, is by creating kind of a generating particles, and then within these things chambers here, we can generate this short-lived radioisotopes for us to use. So giving us that opportunity to locally within the facility generate this radioisotope, and then use that for then doing our making our imaging probes. And I said this is actually manufactured by General Electric, and probably cost would be about £1.5 million or something to use. So we have that available. So it's one of those things that kind of falls almost big science, but kind of maybe not too kind of CERN type, but certainly it has some key expensive equipment. So we will use that, and then of course with that ability, we can generate a whole range of different imaging probes, which enables us to actually selectively image different components of the brain's biochemical processes. So we can actually generate a radiolabeled form of water, and then use that to then actually determine cerebral blood flow, therefore how much blood's actually passing in the human brain. And that's such a powerful technique. Often if you have a stroke, then of course your blood flow is disrupted, is kind of as occlusions, and so it'll enable us to see directly which part of that brain has that restriction in terms of blood flow, and then identify the stroke region. Obviously, the key thing which we all use for oxygen metabolism, and of course, I suppose a good example ongoing right now with COVID, we have the brain low oxygen is often a cause in terms of what we call hypoxia, reduced levels of oxygen, and we can use a kind of a form of radiolabeled oxygen to then inject into the human brain, and then we can actually see the distribution in the human brain and know any regions where it's got lower brain use oxygen than normal, and again we'll relate that to a disease pathology. So there's various things I won't go through, but I suppose if there's one key imaging probe which probably represents I would say 98, 95% of all use, and it's used if you ever see maybe a lorry come to a hospital, is based on this radiotracer for fluorodeoxyglucose, which is a variation of glucose, and of course the main thing about glucose, we use it for energy metabolism, so we can actually quantify in the human brain or different parts of the body the use of glucose. And of course, this is very powerful for in terms of cancer on tumors, because the nature of a tumor, it uses more glucose than the surrounding regions. So what you will see by injecting this, you'll see that kind of like higher accumulation of this radiotracer in a tumor. And so that's used very extensively by the NHS to, in a sense, to detect tumors in the brain, and of course, when I say brain, but other parts of the body, be it lung, be it cancer, be it prostate. And then, of course, in a sense of, apart from being able to detect it, you can potentially see a response in terms of treatment. So it becomes a really powerful technique on that. And so I suppose you could say it's a 3D visualization where we can actually look at a receptor region, and here's a well-known receptor in the brain called the dopamine, and we can see his distribution and actually quantify that. So this is really the real power in how we can bring in selective probes. I call it probes, like radiopharmaceuticals, that gives us the ability to understand the biochemical process in the brain, be it receptors, be it enzymes, or be it biochemical processes. So, but for us to do this, and I say going back to radiochemistry, chemistry being the core, we have to make this on-site. I said with a very short half-life of it. I won't go into the detail of the chemistry, but the nature of it, that we're going to find a way of then getting that isotope, in this case, fluorine-18 isotope or the carbon-11 isotope, into the structure of interest. So once it's tagged on, then of course you can use that to then follow it in the body. And to do that, simply because of this such short half-life, you cannot do normal kind of chemistry process that might take 2 or 3 hours or come back in a day. We have to do what is called very rapid organic chemistry to then incorporate that into the compound of interest within minutes. And that is a very powerful thing we end up doing again, coming back to chemistry, we needed to do that. So of course don't look at the details, but the point I'm making here is that we can do far more sophisticated chemistry, multi-step chemistry, but we try to avoid that because to try and do all that when something's got a very short half-life, you want to do it within an hour or two, purify it, it's a challenge. But if there's no other choice, we can do that, but we prefer not to do this. And in this case, what this is a method of looking at protein synthesis in the human body. So, but how do we do this? By nature of it, we can't apply standard type chemistry methods to do it. We can't use, by nature, if you're doing with something that is can be radioactive, we want consistency and rapid processes to occur. So we will use a kind of automation method as computer controlled. And here's something that is actually manufactured. This is just a schematic of the process, and you can say it's chemical engineering, which it is in a small form, manufactured by GE Healthcare. And this is in a way of enabling us to do basically, in a sense, this kind of processes in a way that which we don't have to actually do it in terms of hand. We can program it, and it can do all the process of liquid transfer, heating the reaction, stirring it, and then transferring it from the very basics of the starting material to finally the purified product that can be used and injected for human form. And we have many of these modules in our lab to enable us to do this. And of course, I won't go through this, but because in a sense, we're doing a chemical reaction. Of course, for any chemical reaction, you have to extract the pure compound of interest. And we use commonly is high pressure liquid chromatography, which is very, like you said, we save here 3 minutes to do this, and we'll take it off and we collect the degraded chemical form to get something that is as pure as possible, usually in the 99% in terms of purity, so to actually to use. So how do we process to do this? And of course, it's quite a rapid process, and we can do it within less than 60 minutes to 150 minutes. And in a way, if you break it down into different stages, we will generate the radionuclide from the cyclotron. We will do the actual chemistry, organic radiosynthesis in its next step. We will then perform the purification, as I showed there, with the liquid chromatography to extract the pure thing. And then, of course, it's going into humans, so it has to be in a form that's acceptable for human injection. So that's similar like saline, so we'll go in that form. And of course, before it goes in, we got to do a quality control, make sure it's the compound that's a right identity, it's the pure form, the pH is right, all the things that makes it acceptable for human use. All to be done within 60 minutes, if it's very short half-life, let's say carbon-11, or if it'll be about 2 hours for longer-lived or fluorinated. And to do this, since we are working with something that's for human use, we do what's called good manufacturing process. And here's someone in a clean room, very much similar to what you might see in terms of a more kind of a pharmacy, or maybe kind of see transistors in a kind of room with a very high purity of air, very controlled. And I mentioned that module here is which we use for it installed in there in a way you can say specialized fume cupboard. And of course, we have all the various things for quality control to confirm purity, chemical purity, radiochemical purity, pH, and of course, even bacterial quality is appropriate. So all those require what is a very much regulated environment. Again, underlying Pinter's chemistry application. So how do we do this? In the course, that's very much what I've just showed you is the end game. We've actually got established with a pharmaceutical to give to humans. But we need to, in a sense, a major area of what I do is developing new ones. And it's a bit like drug development. And this is really my what I call my research lab, the molecular imaging chemistry lab, and one of my research group workers there. And if you want, it's the website. And what we end up doing there is from de novo, from starting things, developing new imaging probes to actually understand disease. And it's quite a complicated process. We do discovery chemistry, we do biological assay, we do preclinical assessment. Before the end, we attain a sort of actually doing clinical imaging. And this very much underlines a major areas of our research in terms of developing powerful imaging probes that will help people to understand a disease process or characterize in terms of early detection, or in terms of actually follow if see a therapy is working and the body responding to that therapy. So in a way, what I'll do, I'll give you a kind of example of an area of which a major area which we work in. And of course, one area is very much in dementia research, which is of course, as probably over the recent years, have become a major area for many of our programs. And I suppose a good thing is, in essence, that many governments are recognizing there's a real need to put in money into dementia research, because sadly, over the last, despite the number of years of pharmaceutical development, there's still no kind of a therapeutic that actually works. And of course, just to give you an idea what it is, we are in a sense, it's kind of global brain damage, and this is probably, of course, the most well-known one, of course, is Alzheimer's, and it's August Alzheimer's very first patient. And what it, of course, it consists of is a global brain damage in terms of over years, kind of decades of the damage, different parts of the brain, which leads to functional impairment, memory loss, and other aspects of what we call and define as dementia. Of course, the most well-known, of course, is Alzheimer's, but there are other forms. It's vascular dementia, Lewy body dementia, and of course, they all varies in terms of the different symptoms, the different times of when they occur, and of course, the different impact. But the underlying theme, and of course, the case is that there's still no treatment for any of those things. And of course, we are actually well known is this thing rising in terms of over 65s. And of course, as you get older, the percentage of it occurring is a bigger percentage. And at present, I don't know if this number is correct. I've done this slide a few years ago. I'm surprised it's probably on the higher than this, but under 800,000 cases in the UK. And of course, it's due to set to double in the next 25 years. And of course, apart from the personal impact in terms of the patient, the family, the carers, is a huge and major economic burden. So in essence, a major area for research, for potentially to try and understand the disease process and come out with therapy. And of course, as I said, we have cell death going on to it. So how do we go about in terms of finding a way to apply imaging for dementia research? And of course, the key to all this and a common thread for what we do is to find a biological target that will be characteristic of that disease. You know, there's something that which we can actually image, we can quantify, and then we can use that to maybe potentially detect disease early, understand the undergoing biology, and then of course that biological target could be a potential therapeutic target. And of course that will enable the drug companies to then assess their drugs against it, and if it is occurring, potentially reduce that. And in the case of dementia or the new degenerative diseases in order, a common feature and a characteristic of all these diseases is the presence in the brain of toxic proteins, which accumulate and deposit, and those accumulations and deposits that results in cell death, and then they result in dementia. So those are really very natural targets for us to, biological targets for us to target in terms of imaging, find compounds that will bind to this and visualize this. And this is three types of the very common proteins that do occur. One called tau, which is present in within cells. One more commonly called beta amyloid, which is present outside cells. And these two are the key ones present in Alzheimer's. They're always present and they cause the damage. While in terms of, say, Parkinson's disease, a different protein is present called alpha-synuclei. And again, they're very much similar in terms that they accumulate in different parts of the brain, and they are toxic, and they cause the brain death. So we really have targeted those as the biological components we want to image, visualize, and enable us to actually understand the disease process. So how do we go with that? And of course, and this is where we can say poor man's drug development in terms of what we do, because we are very much similar to a drug company in essence, that we will want to find a range of compounds or singular compounds which we know can bind to those compounds of those, in this case, these proteins with selectivity that actually can, in the same way a drug might target those to disrupt it, we want to find compounds that can bind to it, and then we use that to then determine it in parts of the brain and to quantify it. So we will generate a range, a large chemical library. That's really as much as we can. And this is a good example of what we're doing this for, in this case, the tau protein. And we might use computational methods, and of course, that's a common thing in silico methods, to do molecular modeling to try and enhance our ability to find a compound. But when we're trying to do this, and of course, the different things about this, we don't really want to develop a drug. We want to develop something that we can use for imaging. So we'll try and incorporate position. We can put a actual carbon-11 isotope or fluorine isotope 18 in there. So these are various factors we're trying to do in this generating a chemical library. So once you've got a chemical library, potentially hits, we bring in kind of, I suppose, a range of biophysical methods and apply those under this. Because what we now want to find, now we've got these compounds, which of those 50, 100 or more compounds which will actually bind to that biological target with the right, what I call affinity, what I mean it binds tightly to those targets in the brain. And more important, just as importantly, it only binds to that target in the brain. It doesn't bind to some other biological process. So when we give that in the human body and we see that in a region of the brain or other part of the body, we know it truly represents that pathology, and it doesn't, it's not seeing something else. And here's an example where we kind of using kind of biophysical techniques, and of course here that's kind of a cartoon of showing the different misfolding proteins in the very small forms, and they get bigger, what we call, so it has kind of a larger form called oligomers and a much even larger form called fibrils. And we will use, here's an example of a compound we designed, and we use fluorescent to try and see how it indirectly, how it binds. The stronger it binds, the higher the signal. So if the compound doesn't bind tightly, low signal, and of course high it goes. And we can use that as an indirect indication of saying which compound will bind to either of these forms, and can use that to then select potentially from that 100 chemical library, 5 or so, which says these are the ones which we want to take forward with. And apart from the, I suppose, almost like qualitative indication of binding, we might do a more quantitative determination of what we call binding affinity. Will give us, it shows that it really does bind to that compound with a high basis, and that's the one to go for. And really to show the kind of techniques which we use, without having to go into detail, in essence, we use all kinds of techniques, which is HPLCs and various things to really to come through. So very much biophysical methods are coming into play for us to understand and take it forward. And then without really going in, this is kind of, I suppose, part 2 of in this case of these proteins. And we really want, now we've shown that maybe 1 or 2 compounds which actually do bind with affinity. In this case, we actually generate in vitro what compounds which are present in the brain. We can actually make it in the test tubes, and we can make it in different sizes. So it can go from a very small size here, which you can represent this form, and to this form, to this form. So we will make it kind of larger sizes to the large kind of long fibril forms, which you know are present in the brain. And then we can use that to then actually determine how well these compounds bind to this form and this form, to really to show which one is it selected for. Then, of course, after that, of course, the main thing, of course, is we're not going to want to use this in humans. So we'll obtain from a human brain bank, we'll have human tissue. And of course, the reason we'll have human tissue is to then show where it binds to, if it actually binds to the natural form in the human brain. The good thing about these compounds, they're fluorescent. So when they bind, we'll see something like this. You can actually see the binding to so this is equivalent in the human brain, and we can see the fibrils. So it really shows it binds with the required selectivity which we want. So before we actually do any human studies, we can actually see it is binding in a way which we want. So that really kind of, from our kind of biological target, our 100 compounds, we potentially now come down to a 2 or 3 compounds which look promising. And so we will then use that, and of course, without going too much into detail, we'll then go through radio labeling of the compound, get the isotope into it. We will then do some further biological characterization, because clearly we want something to use for brain entry. And of course, one of the unique things about the brain from any other parts of the body, it has a blood-brain barrier which prevents toxic chemicals to going in, but also to other compounds. We want to make sure, of course, that our compounds enters it properly. And then we might, just to make sure it's safe for human injection, we will do toxicology assessment to make sure it is fine before we do very well first in human clinical PET imaging, and that will really give us the first indication in humans: is this working? Does this bind to the regions of interest for us? So this is what we'll do, and this, if you could say, is the end game. Here is our combined, in the Wolfson itself, our combined PET-MR. You can't see it all hidden away there. Here's a patient. We'll inject it, of course, an hour of scanning, and we will get something like this, which is here. It's a combined MRI scan and a PET scan. The gray is the MRI, and while the colored shows the accumulation of this radiopharmaceuticals in the different parts of the brain, and that reflects the biological target. And I suppose just to show you in a bit more, I wouldn't worry about too much details there, but what I really want to show here is that we can see the contrast between a healthy human. So here in the humans, injected into humans, very little of it accumulates at all because this toxic protein is not present. But in Alzheimer's, you can see the hotspot present with the characterization. There's a real contrast between that and healthy controls. But also, not just showing Alzheimer's dystrophy, we can look at different, in this case, a different type of dementia, and we can see that it has a different distribution. So not only can we tell the difference between a healthy person with little of it present, but we can distinguish between different forms of the dementia just based on this distribution and quantification. So again, without going into details, we can not just look at one aspect of that disease, we can look at in one patient's different components, the different proteins present, amyloid and tau. And also we can look at inflammation. I think a big area we mentioned about COVID, the big aspects of inflammation driving things, and that's important. So we can actually do individual dissection of the different components of the disease, and it makes us understand how they all interrelate to then have a better understanding of the disease process. And really, this is one of the things that we can actually do more and more so in terms of human pathology. So finally, I suppose just to kind of put you in the context, we do all this work, but it's not standalone. We are very much a part of a wider kind of, you could say, biomedical research, which I showed there on the big campus. So this is, I suppose, the map of the campus, the Wolfson Center there, but we're part of a great program in research. We have a memory clinic where kind of patients who are suffering from various diseases will see the clinicians for that early stage assessment. We will basically have a AR UK Drug Discovery Institute based there who are working in terms of developing the new drugs and to kind of take forward to try and target a disease process. And we also have a kind of a brain bank for that tissue to enable us to actually characterize in vitro in terms of our binding. And of course, apart from our PET technology, we have a different and more powerful MRI scanners present. and something which I won't go into, one called Meg, to really to kind of apply all those range of techniques in an integrated fashion towards kind of in the major effort to understand dementia, and really to enable us to come up with potentially therapeutics to address this major health burden. And so I'll end there. I've said some acknowledgements as kind of members of my groups in terms of the molecular imaging chemistry lab, and the people who make it for actual routine, our routine clinical studies. And of course, shouldn't forget the funders, Medical Research Council, EPRC, and GE Healthcare, who kind of captures all the work I've shown there. And really, thank you out there in the internet world for listening. Thank you. I'll stop there. Okay.
Okay.
End slideshow. Sergio?
Yeah, I think you can probably.
Okay.
Great. So thank you very much, Franklin. That was really interesting. So it seems we have one question already from an attendant about the lecture. But I just wanted to remind everyone who's listening that we'll be moving on to a Q&A about admissions in the natural sciences after this. And so if you have any questions ready, please feel free to submit them through the Q&A feature at the bottom of the screen in Zoom. You can ask questions about any aspects of the admissions process, what it's like to study natural sciences in Cambridge, more general questions about Cambridge. Okay, so, but why don't we answer the first question? So somebody asks, since PETs are still relatively expensive nowadays, how could we reduce the cost to access these technologies?
That's a really good question, and I really could say that's one of my hobby horses. In essence, to get PET from what it is now, very much centered at major biomedical research centers like the Addenbrooke's or King's College Imperial, that can enable us to, in a sense, to be used more widely, to be used for kind of routine, like a standard nuclear medicine technology. That is really what we want to drive to. And I think the way to do that is really going to be in terms of the technology. We're going to bring in terms of, I showed chemical engineering. Instead of having what I showed there is a large module with a large fume hood, bringing things like microfluidics, bringing kind of new technology to make it more compact, and using methods that just generally make it simpler, more robust, and more reliable. And really, by doing that, it's a way to really to make us to drive down these techniques and make it more widely available. And that's really it's an area which, for many years, I really think we need to address to really simplify, to become, I said, to be kind of a standard nuclear medicine technique that can be widely available. The scanners, of course, is something else, but the scanners aren't, not uniquely different from, say, MRI. So that's really not the challenge there. It's really the end which I've showed there, the quite complex chemistry of cyclotrons, large radiopharmacies, and that drives the cost. And one of the things is to make those compact. And I think the technology is coming through, more smaller cyclotrons, more ideas of new improved chemistry techniques, and that will take us away where I think would potentially be a cheaper, more widely applicable method.
Great. And I think something that I find very interesting is that the fluorine-18 compounds can be used, they can be shipped further since they last longer.
Absolutely. Yeah, that's a very, very good point. Absolutely. One of the drivers, we often start with very much on research level with the carbon-11, which I mentioned, I got 20-minute half-life. So by nature of it, they can only be made one batch for one patient. That cost is a cost itself. The beauty about fluorine, it's a 2-hour half-life. You can make a batch, it could be used for 5, 10 patients, And apart from in making it, say, in Addenbrooke's, and which a good example, which we are doing, you can actually ship it to London. And we are doing that right now. We are shipping it to London. And that enables what hopefully will be, talking about cost, potentially creating regional pharmacists, regional pet pharmacies that will underpin a region in terms of shipping kind of radio tracers to different regions. Each center, may say in Norwich, in Ipswich, have their own PET scanner, and then in a sense, they have one place that supplies it. And again, that will certainly reduce cost and access to this technology. So that's a way, again, we are moving towards for many approaches.
So we have one more question. It says, thank you for the great lecture, sir. I understand that PET is mainly used to diagnose neurodegenerative diseases, but there is not a treatment available yet. How will PET technology help find the treatment in the future?
Well, I'll break that down. The main use of PET is mainly still in kind of cancer, clinical diagnosis for cancer, for staging cancer. And that's where it's in the clinic right now. And still that represents the major aspect. Absolutely, in terms of new generation, it's coming a major area on that. Clearly, in a sense, there's no treatment right now, but I suppose we are very hopeful of this Biogen thing that came through 6 months ago. But what will enable us to do, which we can't do right now, is to understand the biology. We know that these toxic proteins are the underlying cause. How they cause the damage to the brain. We understanding more, there's more inflammation to understand how it occurs very much. But just as important, in terms of clinical trials, for many years, you have the drug companies, trying out different kinds of drugs, and they have a cohort, a group of patients with Alzheimer's. Sometimes you can't be sure that actual person had Alzheimer's. So you're giving the drug to someone and potentially might have Alzheimer's anyway. So what imaging can do, and which is actually doing, is selecting those patients and confirming that this person you give this drug to target this biological disease actually does have that disease. So that's a really important thing to enable us to have much better and much focused clinical trials. But finally, it does appear in terms of a therapeutic this way, and I showed a good and a potentially good example with the Biogen one about 6-9 months ago. They actually did see that drug making a change in terms of the protein. So the early before you see the symptoms, you might see that change in the symptoms. So that's where the power of this kind of technology can really help towards this.
Very well. So thank you very much again, Franklin. And I'm sure that if the audience were here live, you would get a big round of applause.
Another cons of the of webinar of Zoom.
It's been wonderful, and I enjoyed seeing the pictures of your lab, which I hadn't seen parts of it before, so it was very enjoyable. Okay, so I guess now we can move on to the second part of the event, which is a discussion of admissions in the natural sciences. You can also ask more general questions, or if you have a burning question about a different subject, you're also welcome to ask it. Okay, so we have already one question, so let me, so it's about whether you need further maths to study chemical natural sciences at Cambridge. Okay, and so that's a question that's very common, and the answer is for the more physical natural sciences. So if you're studying the physics strand in natural sciences in Cambridge, further maths is very highly recommended. For biology and chemistry, it is not necessarily as critical, but it is still recommended. It is not, I mean, we are aware that not every school in the country offers further maths, and so not everyone will be able to take the course, and so it's not a requirement. What may happen is if you haven't taken mathematics or further mathematics, in your first year in Cambridge, you would be required to take a maths course as one of your elective courses. So this, this is actually a very good opportunity. So before we answer more questions, I just want to highlight a few resources on the web that I think everyone who is intending to apply to Cambridge for the natural sciences should see. So I'll just share my screen quickly. So I think you should be able to see my screen. Let me know if you can't. Can anyone confirm that my screen is visible?
Yeah, we can see it. Thanks.
Thank you, Alex. Okay, so so the first page that I'm showing is just you can access it by going to undergraduate.study.cam.ac.uk/courses/natural sciences. And it's also the first hit on Google if you Google Cambridge Natural Sciences. And this page is critical. It has all the information that you need about what it's like to study, so what the course is like. So, you'll have an overview tab here where the course is outlined. You have an outline of the course structure year by year. Okay, so natural sciences has a very specific structure in Cambridge. And one of the differences between Cambridge and other universities is that, at least for the first year, the course is very broad. So you'll be taking courses on a broad subject, a broad selection of science subjects. So, in other universities, you might apply to study chemistry or physics specifically from the first year, whereas in Cambridge, you are a natural science student. But most importantly for us today, we have a list of entry requirements. Okay, and so in these requirements, you'll read an answer from about the A-levels requirements. Okay, so it says that most students have at least 3 science or mathematics A-levels. The minimum requirement is 2, but this will restrict your choice in part 1A. Okay, and it says that some useful combinations are biology, chemistry, and maths or physics, chemistry, maths, and biology or physics, or physics, maths, and further maths. So this last option would apply mostly to people who are studying the physical natural sciences. And it says that if you don't have A-level mathematics, you're required to complete some preparatory work before the start of the course, and you must take mathematical biology as your mathematics subject in year 1. Okay, so it is recommended, but it is not absolutely required. You'll also find these 2 files. So the first one I have open here, and these are requirements, the typical entry requirements, or the typical offer for each college in Cambridge. Okay, so if you're curious about how different colleges differ in terms of requirements or typical offers, which ones require an A-star, A-star A, or just a single A-star and 2 A's, you can read about it here. So this is for the physical natural sciences, and there's a file for the biological natural sciences. And there's also information about something very important, which is the admissions assessment. Okay, so those of you who are already preparing your application will know that there is a pre-interview written assessment for natural sciences in Cambridge. And this is something that you must be registered by October 15th, which is the application deadline. And it happens at the beginning of November. And you have a lot of information here about the admissions assessments. So this file here, which I have opened in my computer too, sorry, I can't seem to access the file without hovering over the Zoom. Apologies. So but if you open this file, you'll have a lot of information about the admissions assessments for natural sciences, and most importantly, you have a very long list here of sample papers from previous years and specimen papers for this year in particular. Okay, so there are plenty of resources here to practice for the pre-interview assessment. And finally, I wanted to highlight a couple of resources in Cambridge that will be useful. So one of them is Isaac Physics. So this is a website that is designed by natural science lecturers in Cambridge. And it has a lot of information. So it has supplementary material that you can use to supplement your A-level courses in physics. So these will be, here you'll find a selection of problems that just have a bit more difficulty perhaps, or they have a slightly different style from what you may be used to in school. And they're a great way to prepare for the pre-interview assessments, for your interviews, and also for your A-levels. And finally, there's Enrich Maths. I don't know how many of you have heard of this before, but it's another enrichment program that is designed in part in Cambridge. And here you'll have, this is focused on mathematics, but it has many problems that are specific to mathematics in chemistry, mathematics in biology. So if you feel that maybe your preparation in school in maths, in chemistry, say, is not completely adequate, or you're lacking a bit of confidence there, then this is a great resource for you to check out. Okay, so it can be a great complement, a very stimulating complement to your regular course in school in maths. Okay, so I just wanted to, I'll stop sharing now, but I just wanted to highlight those things because you may find answers to some of the questions, and if you forget about our answers here, you can always look at these resources later. I don't know if there's anything that any of our panelists would like to add on this issue of maths or further maths.
Yeah, I might say something just about the biological natural sciences. So even though it does say maths is not a requirement, large majority of the people that do biological natural sciences have all done maths at A level as well. But again, as you said, everyone does the mathematical biology course in first year, so everyone's at the same speed anyway. So I wouldn't worry if you haven't done it, but if you're kind of choosing between whether doing maths or not, I think it would be a good idea because a lot of the options do involve maths when you get here as well. So yeah.
Yeah, and then from my point of view with the physical side, I think you'll find the large majority of physical natural sciences have also employed maths, and of course it's not offered everywhere, but further maths I'd say definitely helps with the first year maths course.
I think we hear a lot of noise with your microphone, Ethan. I don't know if there's some maybe it's not plugged in all the way, or.
1 second.
Can you try to say something? I don't know.
Hello, can you hear me?
Yeah, okay, that's much better.
Okay.
Yeah.
Sorry about that.
Okay.
I was just saying, for the physical side of natural sciences, you'll find that a lot of physical natural sciences have done further maths A-level as well, and that's not always offered everywhere. But even still, it will help quite a lot with your first year maths course if you have done further maths. But I did know quite a lot of people that didn't do further maths A-level, so it's not the end of the world if your school doesn't offer it or if you can't do it.
Okay, thank you. Okay, so so we have another question that says, I want to study medicine, and this course requires a lot of work experience, but due to COVID, this has been very difficult to acquire. But I still have read around my subject as I enjoy it a lot. However, what is Cambridge's opinion on work experience? So that's a very good question. So we understand that this year, many people who would normally have spent the summer or part of year 13 engaging in some kind of work experience for medicine, will not be able to do that because of the COVID-19 situation. This is a very unusual year in this sense. So if you haven't been able to do any work experiences in person, you should not be worried about that. And there's a very good resource as well, which is a list of extra or supracurricular activities per subject for different subjects that are taught in Cambridge. And so these are resources that are published by the university as a way to help prospective students get more information about the courses, but also prepare for admissions interviews. And so I'll find a way of adding a link to it, maybe at the end of the talk. And there you might find some alternative ways that you can use to prepare for your medicine application. But yes, we understand that work experiences this year will be difficult. Thank you for the informative webinar. I just wanted to ask if I have to take A-level maths to study pediatrics. Okay, so I would say for medicine, mathematics is not a requirement. I don't think it is required. So if you want to study medicine at Cambridge, you wouldn't have to have taken mathematics. Okay, so we don't have any open questions at the moment. So what I would suggest is, while we get some more questions that may be more specific. Oh, I'm sorry, we have a new question. So it says, if we do have work experience, will it be an advantage? Also, if my personal statement for medicine is quite specific to one piece of science that I like, would that be good or bad? Okay, so that's a very good question. So certainly, if you do have work experience, that will be an advantage, as in a typical year. But really, as I mentioned before, this will be with the understanding that many applicants who would, under normal circumstances, have been able to do work experiences won't have done them this year. And writing about a very specific piece of science in your personal statement, to me, that sounds like a wonderful idea. And that ties in with, what are we looking for in a personal statement in general? So the key thing to remember about personal statements is that they should show that you have interest in your subject, that you have done research in your subject, that you have thought about what it's like to study your subject. And so if you're able to discuss with a level of maturity a piece of science that you're particularly interested in, that's a wonderful thing. It's also important to try to be honest in your personal statement, and because you can always be asked a question about it during your interview, and we want to see the real you. Those are things to keep in mind. But I don't see a disadvantage from writing about something quite specific. I don't know if maybe Ella and Ethan can give us a perspective, and maybe Alexander as well, since he has a lot of experience with personal statements.
Yeah, I mean, oh, you can go, Alex.
Ella, you can.
Okay. I think I'd say with the personal statement, and I think also just in the process in general, don't try and kind of fit like a certain mold of what you think, like the personal statement that they're going to want to see. Because I think if you're thinking about being unique, as some of the questions have been kind of saying, and standing out, you've really got to come from a place of why do you personally like science? And if you're so bothered about trying to make a perfect personal statement that Cambridge is going to like, then I think that takes away from that uniqueness that would make you stand out as well. And I think one way you can probably do that is just trying to link all your experiences in like a coherent way throughout the personal statement. So when you're going through year 12, whatever, just trying to doing as much things outside your subject as well. So when you come to actually writing that personal statement, you've got a lot of things to choose from to make that kind of link when you're going through it and define you as a candidate in science. So yeah, I think that'd be good.
I think that's very good advice. Yeah. Alexander, I think you were meaning to say something, maybe.
You know, it's totally fine to write about areas that you're interested in, and I don't think this was for medicine. I think for medicine, you should definitely be talking about science, and you should be going into depth. When I'm checking personal statements, my rule of thumb is that if I'm a humanities grad, I can understand the level of science that you're talking about, that it's not detailed enough. So I'd advise you really to go to town with like all the detail about science, as long as it fits within the character limits, of course. But really, you know, show that you've got that in-depth knowledge about the area of science that you're interested in. And I think that really makes for a good medicine personal statement, especially one that's you know, aimed towards Oxford or Cambridge. I mean, of course, you're going to have as well some people who ask you about sort of marrying it with any sort of more vocational experience. You know, some people would have managed to get something before COVID. And my advice there would also be just try to weave any science and experience that you've got, if that's possible. It doesn't have to be, but I think it's a pretty good strategy to make sure that you're basically satisfying the criteria for both Oxford and Cambridge and for the other medical schools that you're going to apply to. That's my advice on adding science in your personal statement and how to do so.
Very good. Thank you. So let's see. So we have a few more questions. So someone asks, what makes a student stand out so that they can be considered as a Cambridge student? And we have two current Cambridge students here, so maybe they can weigh in on this. I would say that there's not a single type of Cambridge student, right? It's an incredibly diverse place, and you know, people have all sorts of intellectual interests and backgrounds. So, I mean, I think from the point of view of an application, we're looking at students who have shown to have the potential to do well in Cambridge, okay? And that means having done well in their GCSEs, people who have good references from their teachers, who are taking their A-levels very seriously, and who have an interest in their subject, okay? So, you know, when we talk about supracurriculars in Cambridge, we're not talking about, you know, whether you've taken dance lessons or, you know, it's or you go hiking, you know, we're talking about things that are related to your subject. Whether you have tried to go perhaps a little bit beyond what is taught in your school, and you have looked for resources in your subject, whether that's medicine, whether that's biology or physics or chemistry, to inform your choice of a university and your university applications. Is there, I don't know if there's Ethan or Elle that could add something to that.
Yeah, so sorry if my audio is a little bit off. Can you hear me fine?
Yes.
Okay, my internet's just a little unstable. Yeah, just to add on that, I think that a lot of students with the personal statement get kind of worked or interested in their subject. Actually, it's you know, you can think quite creatively about how to get that passion for science across in your personal statement. It's not always about reading a book or attending a lecture by a certain professor. You know, you can watch YouTube videos, you can do an online course, you can learn a new programming language, you can give a presentation to your class. You know, I think it's as long as you're, you know, really committed to exploring and reading around the subject beyond your A-level studies and actually being able to express that across in your personal statement, it shows that you actually do care about what you study and you want to do that at Cambridge or whatever university you're applying to.
I think that's very good advice. Yeah, there's so many ways of engaging with a subject these days. You know, even like watching a YouTube video, or maybe you've worked your way through many problems in one of these websites that I named. You know, like Isaac Physics or Enrich. That's the kind of thing that admissions tutors really appreciate when you talk about not only necessarily reading a book on popular science, you know, say a book by Stephen Hawking, or having attended a lecture, but showing that you care about the nitty gritty. So maybe I looked at some problems for the Cambridge admissions exam, and I worked in the summer. I tried to work through, you know, 3 or 4 exams, and I found these particular challenges in the problems interesting because of X, Y, and Z. You know, so there are many ways of engaging with your subject, and they're all valid.
Yeah, I think that, yeah, the same. And I think hopefully, I obviously appreciate it's really hard at the moment, but hopefully these kind of the courses that were in person, I'm hoping that some of them might be able to go online and things like that potentially, or maybe next summer. But there's loads of other things you can do than the typical summer schools that everyone tells you to do. So I think there's like different essay competitions and things through different colleges, and I think that should still be on this year as well. Like lectures like this, and you can talk about these in your personal statements. So I wouldn't get so worked up about like not having those typical summer schools that everyone talks about, because everyone's going to be in the same boat this year. So yeah, but I understand it is kind of hard right now.
Okay, so I have another question on volunteering I take it for medicine. So what alternatives could there be to volunteering, as it is also difficult to find opportunities in care homes, etc.? Here, I mean, I have to say I'm not terribly familiar with volunteering opportunities in medicine, because my subject is math, so I've never done a medicine interview. I don't know if, Alexander, if this is something that you have, a question that you have encountered at Target Oxbridge.
If you know, at the moment, well, certainly there are certain volunteering things you can do with regards to COVID. So whether it's you know, helping the vulnerable, helping the elderly at this time. There are lots of volunteering opportunities around that at the moment. There's likely to be certainly some volunteering opportunities, I imagine, over the winter as things start to get worse. So keep an eye out for those. They will be adequate experience if you're interested in studying medicine. So even if you're not allowed to go into a hospital, then that can be a really good volunteering role that you can take part in.
Yeah, I mean, that sounds like a very good advice. You know, there are probably many community groups that are doing assistance for vulnerable people during this second wave of COVID. So something to look out for. Okay, so we have a question from Osen. What would Cambridge consider an impressive supercurricular in a personal statement, specifically in medicine? Will there be a greater emphasis in admissions exam this year due to COVID and lack of clinical experience? So it is, I mean, I must admit that this is going to be a very unusual year in admissions. So because interviews will have to be done remotely, in subjects like medicine, yeah, this has affected the typical experiences that students would have, and so it's hard to say that the emphasis and different components will be the same. I think potentially admissions exams might receive a bit more weight, but I think it's important to still try to produce an application that is as holistic as possible. So, and I don't think, if you have a very strong application in one aspect, but perhaps your admissions exam didn't go as well as you expected, that would not necessarily be the end of your application. So it's hard to answer this question. What would be considered an impressive supercurricular in a personal statement? So in medicine specifically, I think anything that you can say to show that you have an interest in the scientific fields that are related to medicine, so in biology, chemistry, or any kind of personal experience that you may have having to do with medical care. Again, so it could be a COVID support group for vulnerable individuals. I think all of those would be very impressive supracurriculars in medicine. So I can show you also a document. This is a document that I'll share a bit later. So this is a list of supracurricular recommended supracurricular resources by subject, and this is in the website of Christ College in Cambridge. And for medicine, there's a list of preparatory readings in biology. You have a link here. Oops, I'm sorry. Looks like the link is broken. There is a medical student handbook that you might find helpful. There's a guide from the NHS on how to find work experiences. And maybe there's some information there about how to do this in this unusual year. There's a link to societies like the British Medical Association, the Royal Society of Medicine, radio programs and medical ethics. There's a link to the Wellcome Trust site, which has many educational materials and having to do with medicine. Human Genome Project. So if you these are just starting points that you can use as ways of enriching your education at school and building a good supracurricular case in your personal statement. So I'll add a link to this file at the end of the presentation. So we have a question from Hanifa who says, so if I'm guessing she's referring to her personal statement and writing about allodynia and hyperalgesia and nociceptor sensitization. Sorry, that's Greek to me. I don't know what it, maybe Franklin would know more about it. But I think any writing about a specific scientific subject, whatever it is, if you are genuinely interested in that subject and you've spent time coming to learn it, I think it's a great thing to write about in your personal statement.
I didn't catch the details of what particular items you mentioned, but I think you captured what your response was. Any statement about a particular area you got a real interest in and can really bring some unique insights will certainly be of help, and it adds to the case of why you are interested in doing this. So I absolutely agree.
So someone is asking, I want to study mathematics. Okay, great. So that's my subject. Hopefully I can be helpful there. What kind of things should I include in my personal statement? What would make me unique? Any examples of things that I could add, etc. So again, here in maths, we're very interested in getting evidence that you have tried to approach problems that are a little bit beyond what you would see in a typical school education. So in maths, as you know, we have an exam that happens after you get an offer. It's called the STEP exam. And we have a lot of information about it on the web in Cambridge. And so, for example, if you mention in your personal statement that you have been thinking about STEP problems, or maybe problems that you found in the Enrich website that I showed earlier, or maybe that you thought about problems from the United Kingdom Mathematics Challenge. Those are, and perhaps if you can talk a little bit about what aspect of a problem sparked your interest. Maybe you learned about the method of induction and you found a proof using the method of induction particularly interesting, and that sparked your interests in math. So that, I think those are things that really stand out. So many applicants will list a number of things that they've read, perhaps books on popular maths or YouTube videos that they've seen that discuss different math topics, and that's great. But what makes a personal statement really outstanding is if you can describe a specific detail about one of these things that you've read, a problem that you've approached, and why you found that interesting. Okay, and maybe you don't have to show that that you know exactly what part of math you want to study, because we understand that when you are in year 13 in school, you won't be familiar with university level maths, but we want to see that you have engaged with problems that are maybe a step closer to university level maths, and that you've thought about what makes maths interesting, what makes proving a theorem interesting, what makes solving a problem in say mathematical physics interesting. Okay. I don't know if there's anything maybe Ethan would add to that.
Yeah, that was a pretty comprehensive description, I'd say. I suppose a more science perspective on that, just some of the things I guess we were talking about earlier, and I know that you mentioned looking at the natural sciences admissions assessment for some maybe part of the questions, and what about those particular questions or maybe the topics behind them actually inspired you to look further into the subject, and doing things like Isaac physics in your own time, again just reading around the subjects to really show them that you do want to study that subject at university and you're doing all you can to actually explore that subject in your own time.
Great. So we have a few questions, so maybe, and I'm aware that we're nearing the end of our time, so maybe I can see if there are some common themes that we can discuss. Okay, so one question would be, what work experience would be good for chemistry and biology in the natural sciences? And I think for natural sciences, work experiences they're not as important as in medicine. So in medicine is particular in that it's something that we seek specifically. For the natural sciences, I think we're mostly interested in academic supracurriculars. So, having done, obviously you need to have strong GCSEs, and you need to show some interest in the subject. What grades do typical students who apply to study medicine at Cambridge achieve at GCSE? Those kinds of questions, I think they're best answered through the website that I showed earlier. I showed the website for natural sciences, but there's a similar website for medicine that will have some information on that. Then I can't say that there's a threshold for GCSEs because we look at many components in the application, okay, and also what kinds of GCSEs people have achieved in your school specifically. So we evaluate students, to some extent relative to the average scores in their school, because what we're trying to do is evaluate potential, and not just how many 8s or 9s you got. So someone's concerned about the GCSEs having been marked very differently this year. So I assume this is someone who just finished year 10 and is starting year 11. And this is a difficult question to answer. I mean, the truth is we don't really know yet how we will be considering this year's GCSEs. It is possible that we will look at A-level predictions. We'll give them a bit more weight, but it's very early to tell at this stage. So I completely identify with the anxiety on this. It's a terrible thing what happened this summer with GCSEs and A levels. And so I would say my advice to you would be to focus on your studies, try to do as well as you can in your A levels, in your sixth form, and try to enrich your education with as many extracurriculars, supracurriculars as you can, and eventually we hope to provide more specific information about GCSE requirements. Hanif asks, what happens if we go into another lockdown? Again, this is completely understandably causing a lot of anxiety for many applicants. And I can just reassure you by saying that we are considering very many eventualities as we plan for this year's application cycle when it comes to interviews or pre-interview assessments. And so we're doing our best to make sure that everything happens fairly, but it's very hard to describe in just a few words what would happen in another lockdown. And that may vary it may depend on your subject, because some subjects have different types of assessments than others. How much does Cambridge support you to help you find a career path while you're doing your degree? So that's a very good question. Maybe Ella and Ethan can comment on that.
Yeah, there's definitely a lot of support for career, and I think, yeah, a lot of people say that they're really grateful for the support they get. So for example, there's the careers service, which has like events throughout the year and like fairs you can go to talk to people about careers, and just generally being surrounded like so closely by academics. For example, your DOS, which is your director of studies, and then your tutor as well, just gives you the opportunity to really just ask people about their career trajectories, and yeah, get things such as internships over summer in the Cambridge labs. So there's basically a lot of possibilities when you get here, and yeah, I'm really grateful for the support that I've got with regards to careers and everything.
And you do have to sort of you are independent at uni, so you do have to be proactive and seek these experiences and these opportunities yourself. But as Ella said, the opportunities are there, and it's a matter of actually applying for things and spending time working on your career. But there are plenty of opportunities, and the career service do help you with that sort of thing. And you can always speak to someone with more understanding in what a career could look like in your subject, if you're really lost. Or I know a lot of people at uni that still don't really know what they're doing, or don't really know what they want to do after they graduate, and the career service are there to help you if that's the case.
All right, so seeing as we unfortunately don't have too much time left, I might spend some time after we finish answering some of these questions by text. But I would like to end with a sort of a more general discussion from our student panelists about what it's like to be a student in Cambridge, and particularly a BME student in Cambridge. And so I'll ask maybe each of you, and I'm sorry to put you on the spot because I didn't prepare them for this, but if you could say, maybe describe in one sentence what it's like to be in Cambridge, what your journey has been like being in Cambridge. And second, what advice you would give to your 18-year-old self, or 17, 18-year-old self as you're preparing to apply to university. So maybe we can, who should I punish first? Maybe we can start with Ella.
That's fine, yeah. I think, well, maybe not a sentence, but I think maybe unpredictable. And I don't mean that in a negative sense. I actually mean it in a positive sense. When I was applying to universities, I actually applied for biochemistry at a lot of different universities, and natural science was Cambridge was the only one I did natural sciences for. And in first year, you choose 4 modules. And so I did like chemistry, biology, and maths. And then I just chose evolution and behavior like on the side as an extra one. And it actually just became like my favorite module. And we get to go on a couple of field trips with that course as well, and just it kind of opened my eyes to ecology and conservation. And yeah, I just did not expect myself to kind of go into that field when I came to Cambridge. So that was, yeah, obviously a positive thing. And I think on the BME student kind of side of things, I definitely kind of was surprised when I did come to Cambridge, just looking at the natural sciences cohort, at the lack of BME students there. And I think that is something that should be not like, it shouldn't stop you from applying, but I think you definitely need to understand that that's probably going to be a thing when you do come here, just looking at percentages and things like that. But I don't think that's necessarily, as I said, something to stop you, because there definitely are students here and there definitely are numbers increasing. And there's lots of programs in place to increase BME representation, both in the staff and students. So I think, yeah, it was strange, but having like such close communities, say with the ACS or like the BME campaign, there's so much stuff going on that you do feel definitely part of something. But I think coming from like a multicultural school in London, it was different, but you kind of adjust to that and also become part of the solution, almost in encouraging people to come as well. So yeah, I think that's my summing up of my first 2 years. Yeah.
Thank you. That's...
Yeah. From my point of view, if I summarize it all in one word, I guess I would say like overwhelming a little bit. I think just moving to uni anyway is just a big lifestyle change, and just having to, you know, live independently and just do as you please really, and also, you know, make sure you're keeping on track with your studies. You know, you don't really have anyone looking over your shoulder like you did in school or sixth form, and so it's all really your responsibility to make sure you're on top of things and that you're also enjoying yourself. I suppose a piece of advice I would give to myself is just to really take all the opportunities that you can get and say yes to everything, because, you know, there are a lot of, especially when you get to Cambridge or any university, there will be a lot of societies that you can join, a lot of sports clubs, a lot of friends you can make, and just, you know, having all those opportunities available to you just means that you can make your uni experience the best possible and get the most out of it, because you want to look back and say, you know, you enjoyed your time at uni and you did your best and you made the most of it, and so that's kind of what advice that I would give, I suppose. And in terms of being a BME student, as Ella said, you know, there's a lot of great communities, and the independence of uni means that you can, you know, surround yourself with similar people to you, or people of your color or race or religion, if that's what you want to do. You know, you don't have to you know, associate yourself with other people if that makes you feel uncomfortable. And there's, you know, you'd always find that, and as Ella said, over the years it's getting, representation is getting better and better, but as we said at the start, there is no one type of Cambridge student. There's this, you know, the stereotype that you have to be a posh middle class white man to go to Cambridge, but that you know, that stereotype has completely changed, and it's just not really true, to be honest. You'll find all sorts of people from all different nationalities and speaking all different languages, and it's a really great blend of culture and at Cambridge. And so, you know, don't let that, you know, put you off applying to a university like Cambridge.
Yeah, and sorry, I didn't do the advice thing, but definitely just piggybacking on what you said about just going for things. There's a lot of people asking questions here, and understandably, like this requirement or this grade, and honestly, literally just apply, because, you know, as we said, there's not one type of Cambridge student. We barely even know what the individual admissions themselves, like we have guidelines of what they want, but really we don't know what's actually going to make the perfect student or not. And the only thing that is going to definitely stop you not getting in is you not applying at all. And so that comes with both applying to Cambridge and just opportunities as you go through your university experience. You're not going to get things if you don't go and just try for them. And yeah, I think, yeah, just definitely apply, even if you're worried about your work experience or your grades or whatever, because yeah, you just can't predict what's going to happen. And yeah, just go for it because you're good enough to do it.
Thank you. Yeah, I think that's a very good point. And I mean, something that another student said in a Q&A that we had a few weeks ago was, you have, I think, 5 spots in your UCAS application, and that's actually a lot, right? And so you might as well make one of the spots Cambridge or Oxford, because if you don't apply, then, you know, that you can be certain that you won't get in. But, you know, the fact that you're here, that you're ambitious, that you care about your studies, I mean, that tells me that you have the potential to get in, and so it's definitely worth it. We haven't talked about, you know, very much about what it's like to study in Cambridge, but it's a really amazing experience in terms of the level of personal attention you get, the opportunities you have to explore research in your field, especially in the sciences. As Ella mentioned, things like going on field trips, and there are all these opportunities that are really incredible and that are specific to Cambridge, and so I think you really should consider applying. Okay, so well, we've gone a little bit over time, so I suggest that we stop here. Once we stop the formal part of the presentation, I might spend a couple of minutes just typing answers, but if you need to go, of course, I understand. I just wanted to mention that we're going to have 2 more of these webinars, one that is focused more on engineering on September 29th, and one that's focused on maths. So some of you who are interested in math specifically, there will be a talk about this during the half term in late October. So do check out the website and feel free to sign up for those, and maybe we'll have a chance to answer other questions in those sessions. Okay, so I want to finish by thanking our wonderful panelists again. So thank you very much, Ella and Ethan, for joining us today, and thank you to Alexander for joining us and for helping organize the event today. And also thank you to Professor Aigbirhio, whose lecture was really just fascinating, and I hope everyone took something from it. Okay, so best of luck with your applications, and hopefully see you again soon. Goodbye.